Hello everyone. I would like to give a brief introduction to my final project, which is about to looking for where are G quadruplexes in bacteria. So to start with, what is G quadruplexes? So G quadruplex basically is one type of DNA secondary structures. So we all know DNA can form duplex DNA. At some times, DNA can also form G quadruplexes. G quadruplex are formed by G-rich sequences. As you can see here, they are formed by like at least 4 runs of 3 Gs in one sequence. And the G quadruplex structure here, they can form 3 tetrads holding by metal ions. And they have 3 loops. The loop length of G quadruplex cannot be longer than 12 nucleotides. And there are a lot of both bioinformatic and experimental data to support the idea that G quadruplex might play a gene regulation function in human genome. And so now the question we ask is, what about bacteria? And there are some bacteria that are very GC rich. So there might be a lot of G quadruplex there. So, but where are they? Are they in the coding region or in the TSS upstream region? So this is a question we ask in this project. There are 3 steps. Video, there are 3 steps in this project. First step is to clean up all the files. Well, there are 3 files we need to clean up. The first file is the genomic sequence file, which is a FAST file. And in the first package module of this package, which is a clean up files, there's a function called faster parser. So basically, this function takes a genomic sequence file and then read the whole sequence into a dictionary. And then later, we can use this dictionary for further manipulations. And the second file is a genomic feature file, GFF3 file. As you see here, there are many features in this file, but we don't need a lot. We only want to know where is TSS regions or the CDS regions. So that's the TSS is the beginning of a gene. So what we need to do is find all the features that are genes and find their start locations. If the strand is plus, the start is the first number. If the strand is minus, the start is the second number. And this is the file we want. Only have 6 columns: the sequence name, the start of the sequence, the end of sequence, and then the strand of the sequence. So there in the cleanup file module, there are 2 functions to deal with this file. So the first function is going to read all the genes into a list. If there's no genes in this file, it's going to return like a string. It's going to return a string that saying that there's no genes there. And then the second function taking the list from the first function, and then it's going to write a list into a text file. And later we can use this text file to ask for more questions. And the third file we want to deal with is file for after the quad parser. Is the file actually tell us where the sequence and what is sequence of this G4. And you can see here it's very complicated. So the purpose to clean up this file is also actually to get a file like this. File like this, only 6 columns, the sequence name, the sequence locations, and the sequence strand. So later we can actually intersect these 2 different files. And again, in the cleanup file module, there is a function called trim G4 chromosome function, actually to clean up this type of file. After the introduction, we can utilize the Jupyter Notebook to apply our functions from the modules and merge all the data together and for the data visualization part. So at first, we're going to import all the modules we need for this project. So we need pandas and regular expression, and we need matplotlib, and also we need a special like bioinformatics package. It's called PyBetTools to deal with genomic features. And then we're going to also import all of our modules in this package, like 3 modules here. So HoloView is going to be used to generate an interactive figure. To start with, we're going to set up our base direction, which is a base dir here. This directory is where all of our subfolders and files are. So the first step is to clean up our genomic feature files by the function here. And as you can see here, not every genomic features have genes in them. So we have like a lot of them. They are not well annotated. And the second step to clean up the G-quadruplex location files, and which is the function here. And then we're going to use our the designed class to analyze our genomic sequences. So this designed genomic class, it has so based on this class is inherited from default dictionary. So it has, in this class, it has a property of the sequence and a GC percentage, also the genomic length. And the next class is inherited from this genome class. And it can calculate the G4 density and also calculate the most frequent G-quadruplex sequence if we give them all the sequences in the genome. So if we load all the genomic sequence from the dictionary and also give them the paths of the G-quadruplex location and sequences, it's going to generate all the GC percentage information into a tuple and then later we can read the dictionary of tuples into a data frame, and then as shown here, it has all the genomic identifiers, GCA number, the GC percentage, genomic length, and also most popular G-force. So this is the first step. We have basic information of every bacterial genome. And next step is to find out, well, for every G4 in the genome, how close, what is the distance between the G-quadruplex and the closest TSS. So this can be done with a function in the GFF G4 functions. So there's a function here called get G4 distance to closest TSS. Basically, this function it calculates the distance between the G-quadruplex and the TSS, the closest TSS. So this distance might be 0 or might be minus, which means it's upstream of the TSS. The plus means it's downstream of the TSS. So it's going to return like a pandas data frame as we show here. So I run this function before, save the file into a base directory, and then now I can load this file and read them into a pandas data frame. As you can see here every genomic every bacteria is a column here, and then we can apply all the pandas tricks to generate an interactive figure here. So here, this drop-down menu is the name of the other bacteria, and then every single bacteria has a distribution of the G-force relative to the location of TSS. As you can see, the 0, there's a tall bar there, means it's enriched at TSS. So it might indicate some functions of the G-quadruplex in the bacteria. And then this step is done. So later we have other functions. We can calculate like how many G-quadruplex in the coding regions and how many G-quadruplex actually in different windows of TSS. So I'm not going to go through it step by step, and it's too much. So by the end, we're going to get like this huge data frame and have all the information in it, and like telling us how many G4 in the coding regions and how long is the coding region. Also, how many G4 counts just right at the TSS. So with all the information, we can use pandas to plot data on different columns. Here, the one I plot is group the data by genus. And then we're going to plot the fold enrichment of the G-quadruplex at TSS. As you can see here, the genus Deinococcus actually is highly enriched for G quadruplex at TSS, while the other, like the Thermus, is not. I think, yeah, that's it.